So I guess I'm going to change gears a little bit here. So as a drug designer, when I learned that the contemporary clinical trials with psychedelics were beginning with psilocybin, the first thing that came to my mind was, well, why psilocybin? There are so many different psychedelic drugs. Why start with psilocybin? And so as it turns out, there are quite a few salient points that lead us to using psilocybin and as a potential. Sure, as a good choice. So as Pancho had mentioned, familiarity, extensive history of safe use. This is a drug that has been in development for thousands of years. So we kind of knew what to expect with psilocybin. The duration, so for 5 to 8 hours for a psilocybin experience, while not exactly ideal, and I imagine the clinicians could probably comment on this better than I can. I think 8 hours kind of ties up a whole day, and from an efficiency standpoint, this may be a challenge, but it's not DOM or something where you may be tied up with a patient for 24 hours or more, which could be something of a challenge. Minimal baggage, and basically it's not LSD, so for the uninitiated, just the mere mention of LSD conjures up images of Burning Man and whatever else, not realities. And so, psilocybin, I think prior to a few years ago, it wasn't even, I don't think people could even pronounce it if they saw the word written on paper. I think many people are actually just beginning to learn how to say the word psilocybin. So there wasn't really a whole lot of baggage attached to this molecule. And finally, the anecdotal and preliminary clinical efficacy. So the anecdotal efficacy has been known for some time, and then the preliminary clinical trials have also shown that this may very well be a quite effective molecule. So on the right there, also Sandoz had made psilocybin in the 1960s, and so there was some clinical use with psilocybin, but that kind of went away in the 1970s with the Controlled Substances Act. But it is a molecule that we're quite familiar with. But what about everything else? So I just made this slide from memory. This is maybe 20 or 30 structures, but if you scan through PCAL and TCAL, there are literally hundreds of known psychedelic molecules. So what about all of the others? So yeah, the research arm of the Usona Institute, we've set one of our key goals is advancing scientific understanding into the mechanism of action of these different psychedelic compounds, and ultimately towards the end goal of accelerating the development of new therapeutics. So just figuring out how they work is not enough, because why are we doing this? And it's to try to address and help as many people as possible. So I'm not going to get in too far into detail here, but the gist of this is that there is a lot that we don't know. So on the left side here, this is kind of the contemporary understanding of how psychedelics works. This is basically what 99% of pharmacologists would agree on with psychedelic drugs. So that's the 5-HT2A receptors. This is the protein on the surface of a neuron in the brain. Your psychedelic drug, it makes it to the brain, it binds to that receptor. It causes a pathway to get activated inside the cell. This causes a bundle of neurons to start firing, and you get psychedelic effects. And while this is a perfectly valid explanation of a part of the process, it is very likely much more complex than that. So on the right side, so this is kind of a new paradigm in pharmacology called network pharmacology. So all psychedelic drugs not only bind to 5-HT2A receptors, but they're promiscuous, quote unquote. They like to interact with all sorts of different receptors at the same time, and different psychedelic drugs have different interactions with all of these receptors, and so they interact with them at the same time, and so you end up with a map, kind of, all of these receptors being activated at the same time, then interact with one another, and so there's this kind of complex thumbprint for different psychedelic drugs that we really don't have an understanding of the mechanism of how this all works. And this is largely because we just don't have the capacity to study this. I think with modern high throughput pharmacology techniques and computers and machine learning, we are at the point now where we can begin to piece together these complex interactions and get a better understanding as to how these things are different. And so I call these the black boxes in psychedelic research. So all of this research, it exists on sort of a continuum. You could probably lump all of the scientific research into psychedelics into one of these 4 boxes. So I kind of live down here on the left side on the receptor or molecular end. I'm making molecules and trying to figure out how they interact with receptors. You got the systems biology, so how do these things affect the brain as a whole, how it works, subjective experience, so you've got your psychologist, and then finally the therapist. And so on one end, you've got sort of reductionism, so that's saying that we can understand the whole by looking at each of the individual pieces, kind of like a lock or a watch. You can take all the gears and springs and put them all together and figure out how the watch works. But then at the other end is a more holistic view, and that says that this too shall pass. That says that we really can't understand the whole by looking at all of the individual pieces. And that's what's so interesting about psychedelic drugs. It's not like a beta blocker or something, this drug that interacts with a certain receptor and it causes your heart rate to go down. It's a very easy system to study. That's why we have very excellent beta blocker drugs. Psychedelics, the self, the consciousness acts on the drug just as much as the drug acts on the person. And like I said earlier, this is a new paradigm in how we actually look at the development of drugs that affect the mind. Y'all know this guy? Who is that guy? Sasha Shulgin. So Shulgin, I've been going with experimental psychopharmacologist for his official title. For those that don't know, Sasha Shulgin, he was trained as a biochemist. He was a very excellent chemist. He took mescaline once and found that to be quite interesting and decided, well, mescaline has this structure. What if I move this methoxy group over here and then take that compound and see how that compares to mescaline, and then repeat that several hundred times, and you have p-cal and t-cal. I mean, this is Shulgin's legacy. And so when we were building our research laboratories in Madison, we were really kind of interested in applying the pioneering aspect of Shulgin's work, but also fitting that into the modern paradigms that will allow us to actually reach as many people as possible in a more formalized way. So Shulgin's motto was make them and taste them, and I can't advocate that here. As in bowling, there are rules, and so while Shulgin recognized the value of this so-called clinical approach, I mean he basically said that you really can't understand psychedelic drugs without taking them. You can't just do that today. So, but you can in a way. What do we have that Shulgin didn't have? So, as Pancho noted, we have the ability to produce high quality compounds under GMP. So compounds with good purity and quality. We have toxicology assays. So, Shulgin's approach to toxicology was, he'd make a new compound and then he'd take a little bit of it. And then he'd take a little more. And if he started to feel bad, he wouldn't take any more. And once again, I can't advocate that approach. So we have assays now where you can get just sort of red flags. So you basically treat a cell culture with a molecule, and it'll let you know this is probably going to be toxic. It shouldn't be consumed by anyone. So we have high throughput pharmacology assays, computational methods. We've got all sorts of animal behavioral techniques, and finally, I think we're moving into an era of favorable scientific, social, and legal climate. So let's see, where are we going from here? And so this is a molecule beyond psilocybin that we're taking through that process now. So some of you may be familiar with 5-MeO-DMT here. So. The picture on the bottom left there, so that's a recently synthesized engineering batch of 5-methoxy-DMT. It's about 60 grams of high purity 5-methoxy-DMT. We've officially filed our IND application with the FDA for 5-methoxy-DMT, and the actual GMP synthesis of this compound is commencing in the coming weeks, actually. So. The first stages for the first human clinical trials, controlled clinical trials with 5-methoxy-DMT are underway. So, And so this is as Bill had mentioned, we're a very new entity here. This is just the beginning. As I see it, Usona is kind of a hub in this wheel. We're currently systematically synthesizing and cataloging all of the known psychedelics that exist and providing them to world-class researchers all around the planet, engaging in all of these activities in science in such a way that really begins to fill in those black boxes on that continuum. It's joining those two ends of that continuum in an interdisciplinary and collaborative way so that we can begin to understand the mechanism of action of psychedelic drugs. And so, with that, oops. So many molecules, the time is now. Thank you. I think we have time.
Yeah, we have some time for questions. Is that right? A few minutes? Good.